My name is Parag Mallick. I'm with the Canary Center at Stanford, and since this is an educational section, I'd like to really keep this pretty informal. So if you have questions along the way, just stop me, shout them out, and just by way of background and introduction, my original training is actually half in computer science, half in biochemistry, and systems biology is really the convergence of those two fields. And so a lot of the work that we do in my group is focused on systems biology as well. My postdoc training was at the Institute for Systems Biology and in Seattle, and so a lot of what I'm going to present is the things that we started developing there. Next slide. Awesome. So just a quick overview of what we're going to talk about today. Just to a show of hands, who here is already doing systems biology? All right, a lot of you. And who here is familiar with systems biology already? Okay. And what about transcriptional profiling? Nobody? Okay. All right. Well, this is good. So my hope today was to really lay a foundation and to start off with the concept of, what is a system, just introducing that, and then moving on to where some of these views came from, really taking a step back in time, a couple hundred years. I think a lot of what we take for granted in biology and the way that we think about problems in biology originated several hundred years ago, and so we're going to start there and work our way forwards. And then moving on, we'll talk a little bit about transcriptional regulatory networks, what they mean, how to derive them, and then how to relate those to protein networks. And then moving on to some very small scale modeling. So there are really two different types of modeling in systems biology. There's sort of large scale top down modeling, starting with really big data sets and trying to model the whole everything. And then there's a completely different style where really focusing on a single pathway or even a handful of proteins and trying to model those in extreme detail. So I'll show you some interesting examples from that. All right, next slide. Great. So just to connect this all together, why are we talking about systems biology at a molecular imaging conference? The goal of a lot of the investigations in molecular imaging are to characterize what's going on. What are the biological processes at work? Where are they right now? And where are they going? Really to ask the question, all better? Great. Thank you. Awesome. Okay, so to understand what is the state of a cell or an organ or an organism, and then to be able to predict into the future what's going to happen. Does that person have a disease that's going to progress? Is that cell going to become more aggressive or more invasive? Is it going to move left or right? And a lot of times, historically, biology has been studied by taking each of the individual components of cells, individual proteins, individual transcripts, individual genes, and studying them in isolation. And the whole premise of systems biology is that these approaches are often insufficient for describing the behavior of the whole system. So a lot of when we think about models, this is just one example of a model, right? This is the sort of classic model of gravity, codified in a pretty simple little equation. And by describing gravity in this very formal way, it gives us a lot of power. It allows us to describe things like planetary motion. It also allows us, to describe how juggling works. And so by taking these seemingly complicated things and boiling them down to formal models, it helps us codify our knowledge and then also potentially to predict behaviors we wouldn't have otherwise. So our goal in systems biology is to eventually get to these sorts of descriptions of how cells and eventually hopefully even organisms work. So really the challenge we face often is, given a set of measurements of different things, either the genome or the transcriptome or the proteome, how do we go from these measurements, which could be made by any number of approaches, could be made by molecular imaging approaches, how do we go from these sorts of things to what's a cell going to do? Is it going to live? Is it going to die? Is it going to move? And how do we describe that? So there's this big hole in the middle of how do we get from really this is sort of genotype to what a cell's going to do, which is phenotype. And that's the role of systems biology. So a really common experiment in physics is the sort of suitcase experiment. So you'll see this in freshman physics classes all the time. Someone will set down, the professor will set down a suitcase and say, all right, predict what's going to happen to the suitcase. And then the professor will kick the suitcase in one direction and say, I'm going to push it like this. And then the class has to guess and say, all right, where's it going to go? Now, if it were a simple suitcase, this would be quite straightforward. If you pushed it one direction, it would follow that direction. But usually there's a trick, is that there's something complicated going on inside the suitcase that you don't know that actually messes with that trajectory. So you push it forwards and instead it moves sideways, or things like that. And so then the question the class has to figure out is, why is that happening? And then try to formalize that. Same thing, if trying to predict how a clock behaves, that's a pretty straightforward modeling problem. You pretty much know the hands are going to spin around. It's not a complicated system. It's really just a machine, a very simple, straightforward machine. And so if we go back in time to thinking about how biology works, this is really the sort of model that people had, was that, you had your cell, in it there were some genes, and depending upon that gene, and just that gene, you could then say what the organism was going to do, whether it was going to be healthy or sick. And so there was this direct link between genotype and phenotype. So this came from a reason. So this sort of reduction to single genes, it didn't come out of nowhere. In fact, I blame Descartes. So in a number of his treatises, he said essentially that humans can be described basically as clocks. That if we take them apart, we reduce them to their pieces, understand each of those pieces individually, then we'll be able to put them back together again and describe how humans work. And you'll notice that a lot of biology for many years has really followed this paradigm. Let's take everything apart, study it in isolation, and then it'll help us understand how to put it back together and how it works when it's put back together. There was a follow-up to this by Leb in 1912 who sort of argued that same concept, that really all that humans are sort of overly complicated machines. We're basically clocks. Everything we put in will get exactly back out again. There's no complexity anywhere. So the systems view looks a little bit different. It dates back a little bit further. Aristotle said, that the whole is something over and above the sum of its parts. So if you take those pieces together, you put them together, you get something that you couldn't have predicted. Linus Pauling said, a life is a relationship among molecules and not a property of any single molecule. Really sort of asserting that it's how these molecules interact and chat with each other that is really what drives biology and life. And Francois Jacob as well, that every object biology study is not just a singular system, but a system composed of many systems. And a lot of these ideas about what a system were really codified in a series of books by von Bertalanffy in the 50s. This was really where this concept of systems thinking came from. And so, just to give you a really concrete example, a lot of what systems biology is obsessed with is context, saying that how things behave and how they act depends upon their contexts. So just to show you an example that perhaps is a little easier to relate to, this is one of my old students, Jenny. When she's alone, she's basically, your average nice undergrad. But it turns out, when you take Jenny and you put her into a system with Brady, she turns into a totally different beast. And then if you take her and you put her into a completely different system, no Brady, you put her into a system where there's a triathlon involved, golly, she turns into somebody completely different. Which would have been very hard to have predicted. So, if I were to show you this particular Jenny up here, you would have had a hard time predicting that this same nice, sweet person turns into this really mean, aggressive person. But it's all context dependent. Same Jenny, different context. And all of us are guilty of this. We're all complex systems in this way. I guarantee that many of you sitting in this room right now are not going to be behaving like this at the dinner later tonight once you've had a few drinks in you. So that's really the concept that systems biology tries to start with. And in biology, the contexts are a little bit different. We have things like, one protein interacting with another protein, and that changing their behavior, changing the way that they act and look. We have things like all the cells interacting with each other, and how that changes having one cell near each other, and them communicating. Okay, so if we want to define a complex adaptive system, just to give us some language to talk about these things, a complex adaptive system is really just a set of interacting components where when you put them together, you get some whole. And there are many different ways. Once you have a system, that's sort of any system. A system is called complex. A complex system if it displays emergent properties. So things again that you couldn't have predicted on its own. Water is actually a great example of this. If you were to study oxygen and study hydrogen on their own in isolation, you'd have a really hard time predicting the properties of water. All of those properties that come from water are emergent properties of a complex system. And another component are complex adaptive systems. And these are systems where they change over time. So your behavior today depends on everything that happened before. There are other systems that are what are called memoryless, and they're completely invisible. They don't care what happened before. So one of the questions we have to look at is which of these different types of systems is biology, and how does that affect how we model and describe it? So some of the properties that come out of systems that we might talk about are nonlinear behavior. So things where we add them up, they behave like something different than just the sum of their parts. Emergent behavior, like I talked about. Self-organization, this is a common property in cell systems that they spontaneously order themselves. Adaptation or evolution, we have lots of examples of that. And then layers of description or nesting, sort of seeing the same pattern over and over and over again. These are all attributes of systems. Okay, I'll skip through that. So the questions we typically ask when we're looking at a system on the first level is what are the pieces? What are the parts? Again, if we were studying a clock, that might be all the gears. For studying a cell, that might be all the different analytes. And then we ask, how do those states change? How do the cells change over time? And then what impacts that? How do we change those perturbations? The most important component is what are the connections? How are they related to each other? They're not just sitting in a box hanging out. They have very specific ordering and relationships. And then we ask about the emergent behaviors and the evolution of the system. Okay. Are there any questions so far? So far, we're just getting some terminology out of the way. So now that we know what a system is, what's systems biology? Well, systems biology, this is a really lovely quote by Edison. To me, systems biology seeks to explain biological phenomena, not gene by gene, but through all of the interactions. To gain an understanding of the mechanisms of how biology works. And a more formal term that came from Lee Hood really casts it in four pieces, saying, all right, the study of the systems biology is the study of the mechanisms underlying biological processes, and it really has four key parts. One are large sets of experimental data. Two are mathematical models trying to explain that data. Three, actual simulation. And then four is assessment of how well your models are doing. And these are really the pieces of systems biology. Okay, so one of the really important concepts in systems biology is scale. We talk about multi-scale behavior. So when we talk about scale, we typically talk about things in terms of length or time. And you're all, as imaging people, you're really familiar with scale and things that occur on the millimeter scale and the inch scale and the human scale. So in biology, we have scales that range from sort of genomic scale all the way up to population scale. And they all interact in complicated ways. There are links within any given scale, and there are also links across scales. In physics, they have the same challenge. They have scales of subnuclear behavior, atomic behavior, molecular, all the way up to astronomical, but they have a significant advantage over us in biology. Their scales are separated by orders of magnitude. It's not often that a subnuclear event is going to in any way affect the rotation of a planet. On the other hand, in our case, in humans, a single genetic change can have a huge population effect. Okay, so again, if we think about sort of the length and time scales, humans really span seconds all the way up to years. And when we're modeling these systems, that's something we have to think about. Okay, So coming back to how do we describe cells working? Well, okay, so this is just saying, we're trying to get from genotype to phenotype, and we're going to have to go through lots of different layers to get there. I stole this slide from last year's talk. I thought it was a great slide, just sort of showing all the different pieces. So we're going to start with our really simple model. So this really simple model came from Mendel. So it dates back to the 1800s. And so everyone's probably familiar with Mendel and his pea crossing experiments. And what he was doing was he was just taking peas and then looking at a small number of traits, so phenotypes associated with those peas. So he was looking at whether they had green or yellow seeds, whether they were tall or short plants, whether they had green or yellow pods, and then some morphologic characteristics. And he would do these experiments where he would take one pea of one type and another pea of another type, cross them together, and then see what he got. And what he found through this is that there were these very clear patterns of inheritance. And it turned out he got incredibly lucky because the traits he was looking at were actually obeyed these very simple rules of inheritance, that taking 1 and 1, you got exactly the right distribution. If he'd looked at something else, he might have found some complications. If he'd looked at genes that had small silent mutations that only kicked in under stress conditions, for instance. But instead, he looked at these blazingly obvious things and uncovered this rule that variations in phenotype were caused by heritable factors. Okay, so far our model's okay. And if we go forwards in time a little bit forward, we actually get to the terms gene and genotype and phenotype, which came out of Morris plant studies in the early 1900s. And so we now had some terminology for talking about these things. And it was later in that same time period that the link between genotype and phenotype, in particular genotype and disease, came about. So that Archibald Garreau in 1909 was looking at the disease called alkaptonuria, and he hypothesized that this came from a single gene change. And so he could explain all of this disease by one gene. He didn't have to consider 25,000 genes. He didn't have to consider transcription or translation. He looked at a single gene and believed that it was related to a single enzyme and caused a disease. So that concept got even more enforced by Beadle and Tatum in the 1940s, who said, all right, well, mutations in single genes can cause changes in metabolic pathways, and they stated this as the one gene, one enzyme hypothesis. So again, we have this concept of, okay, well, we can describe all of biology by looking at a single gene. Our model that says, all right, now we're going to go from a gene to a protein, and that's going to immediately tell us the phenotype and the behavior, is sufficient. Now, are there any examples where this doesn't work? Where I mutate a single gene and I don't get an immediate phenotype like this? Yeah, like almost every gene except for these ones. So they got they sort of, it was convenient, right? So this was their initial model. You'll notice there's a glaring hole in this model. There's this middle layer that we often think about transcription, that actually was not part of the model. That didn't come around till later. So based upon their model, it made sense. If you want to study things, this is what you do. You take your complex mixture, you take your cell, and you purify and purify and purify until you get your single gene or your single protein, and then you study that to death. And then by doing that, you're going to understand disease. Is this going to explain cancer? No, but they didn't know that yet. But this explained all of the things that they'd found so far. Okay, so there was in the 1960s, it's shocking how recent this is, was really when there was this seminal paper in Nature by Sidney Brenner and company that said that they hypothesized this new RNA which links together protein synthesis and DNA. So now they're adapting their model and they're saying, oh, okay, all right, well, there's more to our model than this. Let's put in the RNA. So now what we have is we have DNA going to RNA going to protein. It's this nice, simple, straightforward relationship. There was then some work about Crick's famous paper about the central dogma, saying how do analytes get from one to another in a cell? And this is probably the single most overinterpreted paper in the history of biology. really all he was saying, he was talking about conversion of analytes. He was saying that analytes go from DNA to RNA to protein with, some minor exceptions. He was not saying anything about how cells work. He was not saying anything about how cells are regulated. He was just saying this is the flow of biological molecules. Of course, we missed that point, and then spent many years using this as our model for regulation. So we're going to do that right now. We'll take this model, and this is essentially the model that comes out of that work. It says, all right, well, we have a transcript that leads to a protein, and then that protein then leads to some phenotype, like motility or disease or something. Okay, so this is simple, right? So all we have to do now is understand transcription and how transcripts relate to each other, and then we're good. Okay, well, let's play this out a little bit and see where this goes. Okay, so how do cells work? All right, they work according to the central dogma now. Genes make transcripts, transcripts make proteins, cells behave. So this is sort of the very first study. It was done on this Lac-Operon, a bacterial operon, describing how do cells respond to the presence of sugars. So, okay, that works. So it's all transcriptionally driven. It's driven in response to sugar. It's a nice, simple feedback loop. Okay, so one thing that we have going for us now that they didn't have then is this picture. They had DNA, RNA, protein. They didn't have functional protein. They didn't have metabolites. They didn't have anything about DNA structure or chromatin organization. But what they were thinking about that we need to think about still is where are the control points? If a cell is going to go left or right, it's got to make a choice. It's got to take some information in, compute, and then make a decision. And so really what drives cellular regulation are these control points. And they're different for each gene. Some do have transcriptional control, and so it's sufficient to look at them at a transcriptional level. Others have later control. What we have now that they didn't have back then is we have ways to measure all these things. So right now, there are a wide array of technologies, some for spanning this entire continuum. Some, like sequencing, which are incredibly mature, are already in generation 3. We have other things like RNA-seq, which for looking at transcripts are, shockingly mature. Array methods, incredibly mature, have been around for 20 years. And then we start to get into emerging technologies like activity profiling, in vivo activity profiling, and some of these other more complicated techniques that allow us to look at some of the faster timescale processes. But we can now do this. So, all right, so let's start off with looking at transcriptional regulatory networks. Okay, so one of the challenges that we have, and actually before I go into this, are there any questions about sort of the general big picture of what systems biology tries to do, what a system is, any of that sort of stuff? Is that all clear? Nods? Okay. I always tell my students that I'm not a television and they can't change the channel. So please do ask me questions. Okay, so at this first level of regulation, what we want to understand is how transcription works. So the way that people typically do these sorts of models is they say, all right, well, let's start with some cells, let's grind them up, look at the transcription, and then try to uncover what are the relationships, what genes are co-regulated, what genes are upstream or downstream of each other. And the way that they build these sorts of networks that that look like this, that are basically large hairballs, and say these things are related to these other things. And so the way that they construct these networks is with a series of assumptions. One of the assumptions is that genes with similar expression patterns across a variety of conditions are co-regulated. Another says, and that an edge between nodes, so if I have each one of these dots as a node, the lines or edges, implies a relationship between things. Oftentimes, we will explicitly represent transcription factors as drivers of regulation in these networks because we know that they're clearly important. And sometimes what we find is that a module is required. Our goal, again, is to predict how this system is going to behave. What's the level of an analyte going to be at some time or in response to some perturbation? And sometimes we can't do that based on just a single transcript. We need lots of them in order to connect the dots. And so those are when we pull in a module. So how do we do this? just as a quick show of hands, could all the sort of mathy people raise their hands in the room? We've got a handful. Okay, all right. I wasn't quite sure how much detail to get into in terms of the Greek. So I have a few slides on it, but not too many. So this is one of them. Feel free to glaze out if you're not interested. So there are really three approaches that people use for deriving these sets of network edges. Again, they're all based upon the principle of trying to understand what are the relationships, what things are co-regulated, what things have similar behavior. The sort of originator approach was with what are called Bayesian networks, and these really ask the question, what's the probability of some value of some transcript level being something as informed by some other value? And that tells me something. A more modern approach has been to use what are called information theoretic approaches, which are related and ask the question, how much information does knowing the value of one gene tell me about the level of another gene? So if I have 10 units of gene A, of the transcript of gene A, how much information does that tell me about the number of copies of transcript of gene B? And then the last class are these ordinary differential equations models, and these are really time-dependent models. So if I'm trying to describe the time flux through a system, these are the sorts of models that people use. And there's a really nice review in molecular systems biology. Okay, so just to show you a concrete example of what one of these networks look like, you can see the triangles in this network. This is something we derived in my own lab. All of the little triangles are actual transcription factors. The blocks are clusters of different genes. Any circles you see are individual genes. And essentially what this network is telling me is that, for instance, this cluster of genes is regulated by this transcription factor. And so we derive this network using a mutual information criteria. Don't worry, you're not going to have to look at it very much. But I think a lot of people are surprised. Their intuition about what's going on inside cells is that there are these sort of nice, pretty biochemical pathways. Oftentimes we have a lot of missing information, and the level of connectedness and co-regulation is really quite large. And so this is actually probably a reasonably accurate representation of what's going on inside a cell. And it's incredibly complicated. Everything's connected to everything. It really is a giant, horrible hairball. But even having this is great. It allows us to zoom in. So one thing we wanted to look at was how cells respond to DNA damage. And I don't know if this is going to work, but I'm going to try. Nope. Crap. All right, well, that was a movie. And now I'm totally stuck. Okay, well, if it's possible in the back, could you possibly just click on the center of that? Oops. Okay, well, that's okay. You didn't actually need to see a movie of cells and how they work. But one of the things you can do is you can zoom in to a sub part of the network. And so this is from that ginormous hairball. We zoomed into just a small piece of it that we think is really responsible for how cells respond to DNA damage. And so we treated our cells, and what you would see is each of these boxes, again, are biclusters. These are sets of different genes together. Each of the triangles are transcription factors, and what you'd be able to watch are the dynamics through this network. So 6, 12, 24, 48, 72 hours after treatment with the drug, how are the cells responding? How are the networks changing? And how are those networks changing differentially between different types of cells? So some cells respond immediately, and they have particular factors that come up. Others respond in slower timescales. Some don't respond at all. And you're able to trace those kinetics through there. One other thing you can do is that you can actually do virtual knockouts. So by having one of these sorts of models, again, this is a movie, so you'll have to take my word for it. You can pretend to knock out things. So in this case, we did a pretend knockout of a gene called P53, and then watched the network respond to that knockout. And so by having this kind of kinetic transcriptional model, we can then say, all right, well, if we were to do something, how would that affect all of the other genes? And as you can imagine, these become really tedious experiments, but it's much easier to do these sort of virtual knockouts for every gene and say, okay, I think these 6 are important. These ones cause a huge impact that ripples throughout the whole network. These other ones are pretty irrelevant. Let's not worry about them. Okay, so that's sort of transcriptional regulatory networks. So far, we're at the transcription level. Things are fine. We have our very simple model. It says that we go from transcript to protein to function. Now, how many people actually believe this model? Nobody? Or you're just too shy? I guarantee you that many people implicitly use this model all the time. When I wander around campus, people do lots of transcriptional profiling. They say, oh, look, this thing's up, then they go chase after it. Which is implicitly saying that something being transcriptionally overexpressed, overabundant as a transcript, has some downstream consequence. Okay, well, here's why some of the reasons why that might not match. This is from a great paper in 2007. I know you can't read all of these, but this is just a small list of why the protein levels might not match the transcript levels, including things like chromatin structure and RNA editing, trans-splicing. There are a number of factors at play. So if you actually look at it, so this is a recent paper by Matthias Selbach, and looking at RNA copies per cell versus protein copies per cell. They're on totally different scales for one thing. The other effect is that they actually don't correlate very well. Just to show you one more great plot like this from my own lab. On the x-axis, we have the transcript abundance. On the y-axis, we have the protein abundance. Do you all see the amazing correlation between the two? Yeah. Oops. So, and this happens all the time. I mean, every time we do these experiments and we take cells, we profile their transcripts, we profile their proteins, and we look at the straight correlation and find nothing. And why does this happen? Well, this happens for some of the factors we just talked about before. These things like RNA editing, microRNAs. Anytime you have any nonlinearity in the system, these sorts of things occur. So just to show you some really concrete examples, so here are 4 proteins that I picked mostly at random. In blue is the mRNA level, and red is the protein level. So again, this sort of mirrors what you saw in the scatter plot before. For this one, there's a really nice relationship between the two. The protein level matches the transcript perfectly. Of course, for these other ones, not so much. So one of the things that a number of people are working on is trying to understand this and trying to model this. So one thing that we did was we made a very, very simple model. We said, all right, well, one of the things that's probably going on is that each protein has its own rate of translation and its own rate of degradation. And it's just not, this uniform constant thing for every protein in the cell. So we built in a model where we took transcript levels and then estimated protein levels as a function of a translation rate and degradation rate, which we measured actually for each protein. So once you do that, your model does much better. So just to show you an example from before, so this was one of those proteins I showed you, top BP1. In red now are the transcripts, in black are experimentally measured proteins, in pluses are what our prediction of the protein level would be from the transcript level. So even though there's not a simple linear correlation type relationship between these two things, if you take into account the fact that these are dynamic processes, that each protein is translated and degraded at its own rate, you can recover a lot of these relationships. So it's not that the relationship between transcript and protein is random, it's just not described by our very simple linear model, which argues that we need to dig a little deeper and come up with more sophisticated models. And even this model fails. So here's a protein where we totally failed. If you look at the transcript level and the protein level and our prediction, we're totally wrong. So it turns out this happened about, I'd say, 30% of the time that we were just totally wrong. Now, when we went in and looked at those ones that we were wrong, it turned out they were super interesting. About 80% of them were ubiquitinated proteins. And so what that meant is that in response to the perturbation we were giving our cell, the proteins were actually changing their degradation rate. There was actually a physiological consequence changing the degradation rate. And so our model didn't originally account for that. And so this sort of, again, it was sort of a blazingly obvious thing that people have known for a long time, but it showed us that it was a really, really important factor that we had to add to our model. So, okay. All right, so one other thing that people often aren't aware of, and we weren't aware until we started looking at these things, is sort of the cell-to-cell variation of some of these factors. Working with a group at Berkeley, Luke Lee's group, we looked at a single cell platform based on microwells. And so in this platform, it's basically a lot of very small buckets. And so cells get flown by and then fall into these little buckets. And then by immunostaining and qPCR, you get the protein level and the transcript level for each individual cell for a particular protein. In this case, we're looking at a protein called c-Met. And it allows us to relate the transcript and protein levels on a per cell basis. And you can do this for thousands of cells. And so this is just one example. This is a cell line, a lung cancer cell line called HCC827 cells. And again, if you look at the relationship between transcript and protein, even at the single cell level, it's not really there, right? This is probably about 10,000 cells. And, there's some relationship, but it varies from cell to cell. Now, what was very interesting was from these cells, you can actually make them drug resistant. So we did. We took these cells, derived a drug-resistant population from them, and then did the assay again. And what we knew by Western, by immunoblot, and by qPCR, is that we got overexpression of this gene called c-Met. That was fine. But when we went in and we looked at it at the single cell level, what we found is that instead of what we expected was this whole blob to just shift up and to the right. Because we had an increase in expression, increase in protein, whole blob moves up and to the right. But if you look at it, you don't actually see that. What you see is the blob mostly moved straight up and got a little bit more diffuse. And so what this argues is that our mechanism of resistance was not actually transcriptional. But in a great number of cells, it was probably post-transcriptional. And so by looking at these sort of relationships helps you pin down where exactly the bottleneck is, where exactly the cellular behavior is coming in. So just to zoom in, it's also sort of interesting that there are actually multiple foci, which we're trying to tease out what this actually means. Okay, so, all right, so from all of this, it's clear that at a fundamental level, we don't actually understand these processes, and a lot of work is needed to tease them out. So far we've made it to transcript, we've made it to protein. What about getting to post-translational modifications, phosphorylation? These are clearly key control points in cells. So for this, there are some very nice new techniques coming on the market. So one is what's called mass cytometry, developed by Gary Nolan's group at Stanford. And in this technique, it's basically your normal flow cytometry, but instead of using a fluorescent readout, you use a mass spectrometer, an ICP-MS, to read out different heavy metals. And so you take your cells, you label them with antibodies conjugated to a heavy metal, take the cells, blow them up, and then whatever the elemental analysis that comes out allows you to say, okay, there's X amount of this protein, Y amount of this phosphoform, et cetera, which you can then go and do more detailed analyses. Including what they call spade analyses and things like that. So essentially, they're now building a more complicated model that says, all right, well, our model now has multiple layers and feedback loops. So we have some perturbation that we're making to the cell, which leads to some changes in proteins, makes it down and causes some changes in some transcription factors, which transcribes some things, which then feeds back up to changing the levels of the protein, and the whole thing cycles over and over again, particularly as a consequence of protein modifications. Okay, so far the network I showed you before was a transcriptional network. And now it's really important because there are actually several different types of these regulatory networks, and it's important to keep them straight in your head. So this kind of model has some elements of a transcriptional model, but it also has a protein model in there. So we can actually create a much more fundamental version of this. So this is the sort of signaling network that we're often used to looking at in books that says, this interacts with that and this interacts with this other thing. Again, the transcriptional model isn't that kind of network. It just says these things are related. But what about a network like this? Can we use a network like this? Can we do something functional with a network like this? Well, yes, we can. So essentially, how we can model something like this is as a series, as a very formal network where we have a set of proteins, and perhaps the size of a circle is related to the concentration of that protein, which we can measure perhaps by mass spectrometry or even by ELISA. And then the linkages between these are really binding constants, which we can measure by SPR or some other high throughput technique. And then I apologize for another math slide. In this case, what we can do is we can actually model this explicit protein-protein interaction network as a series of reactions, of a series of binding and unbinding events, where we have each of the components of the model. Okay, so just to show you, for a really simple network, essentially what we have is we have two proteins coming together to form a complex AB that's composed of, and that's related to the amount of free that's in the system and their binding constant. We also have the total amounts of the protein in the system, and we can just solve this system of equations and then figure out at any given time, as a consequence of changing KDs, as a consequence of changing proteins, how is that network reducing to equilibrium. And so this is sort of the most detailed kinetic network you can possibly get to. So the challenges are, of course, missing values. It's very hard to actually measure interaction constants for every single pair of proteins in the cell. It's also, this model doesn't account for complexes that are more than two proteins. So building that in gets very complicated. Another challenge, of course, is that these measurements of interaction strength are often done in vitro. The Aoki group in Japan is doing some very nice two-photon work to measure interaction constants in vivo, but it's very cumbersome. Using things like FRET and some other imaging techniques. And then there's still the challenge of, okay, now I can describe this network and I can describe who's interacting with who, but connecting this to actual functional outputs is really non-trivial. This is really sort of the next frontier of, once you actually have proteins and all this information, how do you connect it to what cells do? Okay, so the last piece, we sort of talked about how there are really all these pieces in cellular regulation, starting over here with DNA structure and moving to metabolomics and cellular localization as factors. And so I just, in metabolome analysis, we started about the genome and moved to the proteome, and the metabolome is going one more level of detail, saying, all right, let's describe each of these enzyme reactions and measure each of the metabolites. And this is really an emerging area of systems biology. People are still trying to figure out how to relate the metabolome and the proteome, but it's a huge area of interest right now. Okay, so just some warnings to be aware of. So one, oftentimes when you do these models, we assume that cells are these big homogeneous bags. We know that's not true, right? Cells have all these little compartments and things moving around inside them, particularly trafficking from even the cell surface all the way into the nucleus. And so our models need to expand to consider all of this variation. On the topic of variation, there are also some other things we have to think about. So far, everything I described, each cell was feeling the same thing. Each cell was the same internally, and they were feeling the same environment. Now, with all of our imaging approaches, we know that there's not spatial homogeneity across cells. In fact, if you look in the proximity of a blood vessel, you have a natural gradient as a function of nutrient availability. And that leads to some variation in how cells are going to behave. Again, that context that goes supracellular is very important. There's also significant amounts of the cell-to-cell heterogeneity. I showed you a little bit in those two-dimensional plots of transcript and protein, but this variation in the evolution of cell populations is also something that we have to worry about in systems biology. Okay, all right, so in the last section, I just want to show you two really little examples of the other side. So everything I talked about so far was taking big data sets, proteomics, transcriptomics, et cetera, and then trying to uncover relationships. The other side of the fence is to say, I'm going to zoom in and look at a really specific system And so, in particular, one common way is to say, all right, I'm going to set up a rules system for positive and negative regulators. I'm going to say that, I have some sets of relationships where they are negative response regulators, that under negative control they're expressed. I'm going to have others where they're under positive control they're expressed. I'm going to understand how these relationships play out in the cell. So one very interesting observation that came out of Jose Villar's group had to do with one of the most overstudied operons in all biology, the Lac-Operon. Now, it turns out the Lac-Operon actually has 3 control elements. It has regulator 1, 2, and 3. Now, it turns out that O2 and O3 are actually incredibly weak repressors on their own. They normally don't, they're normally, relative to repressor number 1, they're barely effective. However, for some strange reason, having them around increases the effectiveness of repression by a factor of 60. So how does this happen, right? I put in my drug, I get some repression if I have O1 around. If I get rid of O1 and just put O3, I get a really tiny amount of repression. If I get rid of O1 and O3 and put in just O2, even tinier amount of repression. But when I have all 3 together, it's 60-fold higher. So how do I explain this observation? How do I model this? Well, so what they said is, all right, well, let's take a look now at the structure of the DNA. So this is a factor I said that we'd mostly ignored in our models so far. And what they did is they looked at these models of where exactly the repressor binding sites were, and observed that having those peripheral binding sites actually changed the entropy of binding. It caused the DNA to fold in a particular way. And that's really where the repression came from. It came from an emergent property of having these weak operators near each other in such a way that they actually bent the DNA to change the structure of the regulation. So I'll skip over this, but the basic idea was that they could then go in and look at the energetics of the system when you had 1, 2, or 3 of the operators, and the only explanation was that there was an emergent property that came from a higher order relationship of where in the DNA were these repressors. Okay, so one other exciting study was done in yeast and looking at how signal transduction networks work, and they looked at a very, very simple signal transduction network that was only composed of just a handful, 4 factors, and said, all right, let's describe how this system works. And so they said, all right, what we want to understand is, where did the system come from? What are the rules? What are the design principles that the cell used to make this network? And the constraints were they needed a system that was robust to noise. If there were variances in the signal they were getting, it needed to tolerate that. But at the same time, it needed to be incredibly sensitive. That's a tough constraint. You need to be resistant to noise and sensitive to change at the same time. How do you pull that off? And then you also need to be able to adapt and recover very quickly and easily. So they looked at 2 models. They said, all right, well, let's try one model where we have just our proteins and then a regulator Y. And then let's try a more complicated model where we have our 2 proteins. We have B undergoing a modification, so you can see it gets phosphorylated, which changes its behavior. And then there's this other protein Z that comes in and is required for modification. And what they found is that and it turns out that each of these different models are codified in different sorts of equations. You can see that in this case they added in this term Z, and here they added in this term for this conversion of B, and they were able to, there's a slide there that shows that it works way better if you use the full model. And so by looking at this topology and changes in the topology and the changes in the equation, they were actually able to understand what are the rules that there must be at this other factor Z that no one had ever seen before. And they were able to then go and fish out and figure out what this Z was. So, okay, so just to conclude, this sort of systems biology, where it came from, really it's an intersection of 2 paths that had been going on in parallel. Their first path was sort of standard molecular biology, improving over time, us learning things, sequencing, getting to the point of high throughput. The second path was really very detailed modeling of understanding systems in incredible detail, modeling them with very formal mathematics, but oftentimes these models were done in vacuo. They were just sort of theoretical pontificating. There was no data to support them. And with the advent of some of the new sequencing technologies and transcriptomic and proteomic technologies, they really were able to merge just over the last 5 to 10 years to become viable. And that's really where systems biology became not just independent biology in one track and modeling in another, but came together. So just some last thoughts. Just to remind you, so what is a system? A system is a set of interacting components that form a unified whole when they're operating together. It's important to understand that oftentimes the behavior of the whole is hard to predict. And then there really are two big approaches. One is this bottom-up, small-scale, detailed models. These are often driven by microscopy. And there's another one which is really top-down, large-scale, driven by omics. And both of them are needed to connect the dots in terms of understanding how cells work, how to reference, how to understand the cellular behavior. And then there's a tremendous amount of work that's still needed and we're still working on. So I'll stop there and thank you all very much and take any questions that I can answer for you.